Assalamualaikum and hi guys! So today we are going to talk about whale sharks. Do you know that whale sharks are actually an endangered species? Yeah, even though they are the biggest fish in the world, they are currently facing a serious risk of decline. The whale sharks, or also known as Rhincodon typus, is a massive filter-feeding shark that mainly eats plankton, and it can travel across entire oceans. Because of their huge movement range, they are really difficult for scientists to track and study.
Even with their size, whale sharks are not safe from traps. They are classified as endangered, mainly due to bycatch in fisheries, vessel strikes, and habitat disturbance. Another big challenge is that they migrate across international waters, which makes conservation even more complicated. As a result, scientists can no longer rely only on visual observations to study and protect whale sharks. Instead, they use molecular techniques based on DNA to better understand whale sharks' populations. In this video, we will explore 3 molecular techniques used in whale shark conservation, which are environmental DNA, population genetics using mitochondrial DNA, and microsatellite analysis. So let's see what those techniques are and how scientists use them to conserve whale sharks.
Let's move on to one of the most modern conservation techniques used today, which is environmental DNA or eDNA. This technique is actually very fascinating because scientists do not need to catch or handle whale sharks directly. Instead, they simply collect seawater samples and analyze the DNA found in the water. Whale sharks naturally release tiny amounts of DNA through their skin cells, mucus, and other biological materials. For example, a study by Meenakshisundaram and colleagues in 2025 used eDNA from seawater samples to detect whale sharks and investigate their population genetics. One major advantage of eDNA is that scientists do not need to physically handle the animals to obtain genetic information, making eDNA an effective tool for conservation monitoring.
What makes this technique really powerful is how the DNA from the water can actually be analyzed to give us scientific information about the whale shark populations. So basically, just by analyzing the water, they can know whether whale sharks are present in a particular area and even learn about population structure. This method is completely non-invasive, so it doesn't disturb or harm the animals at all. It's also very useful because whale sharks are rare and travel long distances, so traditional sampling is difficult to monitor. Therefore, eDNA has become a promising tool for a long-term monitoring and conservation. The second molecular approach is population genetics. Population genetics help scientists understand how different whale shark populations are related and connected. One way scientists study population genetics is through mitochondrial DNA or mtDNA. This type of DNA is inherited from the mother and is commonly used to investigate ancestry and movement patterns. It helps researchers understand how populations are connected over time.
There is a study by Guzmán and colleagues. Researchers examined whale sharks from the Pacific region, particularly around Panama. They compared mitochondrial DNA sequences and identified different haplotypes which represent genetic variation within populations. And the result was clear. Whale sharks are highly connected across large ocean distances. So, even if they are seen in different places, genetically they are still part of one big population network. Scientists can use this information to better understand migration patterns and gene flow between populations.
The third molecular technique is called microsatellite analysis. Think of microsatellite as a kind of DNA fingerprint. There are short repeating regions of DNA that vary among individuals. In the study of Schmidt 2009, scientists collected whale shark samples from different oceans and analyzed these DNA markers using PCR. This allowed them to compare genetic variation among whale shark populations worldwide.
This is where things get interesting. Even though whale sharks were sampled in different oceans, their genetic differences were surprisingly low. This means that whale sharks from different regions are actually genetically very similar. So, instead of being isolated groups, they are probably connected through migration and movement across oceans. Microsatellite analysis is also useful for assessing genetic diversity, which is important for the long-term survival of a species. This is important because it tells us conservation cannot be done by one country alone. Instead, conservation efforts needs to be coordinated at a global scale.
So, when we combine all of these three techniques, we gain a much clearer understanding of whale shark conservation. eDNA allows scientists to detect whale shark through seawater sample without disturbing the animals. Population genetics using mtDNA helps river migration patterns and connectivity between populations. Microsatellite analysis shows that whale sharks from different oceans are genetically similar and interconnected.
So overall, whale shark conservation cannot be achieved by a single country alone. Since whale sharks migrate across international boundaries, global cooperation is essential for their protection. Based on these findings, several conservation actions can be recommended. First, international collaboration should be strengthened to protect whale sharks throughout their migration routes. Second, long-term genetic monitoring using eDNA should be expanded to track population changes over time and support more effective conservation strategies.
Lastly, important irrigation and feeding sites should be protected to reduce stress, such as fishing activities and vessel strikes. With modern molecular techniques, scientists can study whale sharks in a more accurate and non-invasive way.
The more we understand their genetics, the better we can develop effective conservation strategies for these endangered species.